Good morning, good afternoon, and good evening. This is Dr. Wajiha from Getz Pharma, Pakistan, and I'm here with you today from Public Health Line. The topic under discussion that we will be hopefully discussing in this 90-minute talk today is the UK variant for COVID-19. Will vaccines still work? We have a panel of experts today from different countries who would be walking us through this genetic mutation that has come to light in the last couple of weeks and whether we feel it's a serious threat to the world or not. We'll be discussing the different kinds of vaccines and whether this particular variant will have any impact on the vaccine rollout and its efficacy. We've also been hearing that this variant is more transmissible than the previous ones. We'll also be asking our panel of experts this question, whether we should be worried or not, if there are any further precautions that we need to take. And so I'm going to be opening the floor today and introducing each of my panelists one by one. We have 5 panel of speakers with us today. First of all is Professor Zara Hassan, who is a professor at the Arshad University, Section of Molecular Pathology, Department of Pathology and Laboratory Medicine at the Arshad University. Her expertise is molecular and cellular immunology of infectious diseases and pathogen genomics. She's also the president of the Association of Molecular Pathologists. Dr. Zara has been working since COVID-19 started with the government of Pakistan for the COVID-19 response, and she set up the PCR testing at AQUH. She's also currently doing the genomic analysis for SARS-CoV-2 in Pakistan. So welcome, Professor Zara, to the crew. Next, we have with us Dr. Adeel Bhatt, who's joining us from Qatar HMC. Dr. Adeel Bhatt is a professor of medicine and a professor of healthcare policy and research at Weill Cornell Medical College, Qatar. He's the vice chair at the Department of Medicine and a director of clinical epidemiology research unit at Hamad Medical Complex also. He comes with a huge experience in infectious diseases and epidemiology. So I hope that you're able to learn a lot from him today in the session. Thank you, Dr. Adeel, for being able to join us today. Next, we have Dr. Faisal Mahmoud. He really needs no introduction, but for our audiences who are joining us from other countries, Dr. Faisal is the head of infectious diseases at the Arshad University. He's an associate professor at the Department of Medicine. He very recently received the Tamgha-e-Imtiyaz from the government of Pakistan for his efforts for COVID-19 pandemic in Pakistan. He's been leading the National COVID-19 Task Force. He's also a member of the WHO Working Group of Treatment and Prevention for HIV for the Imro region for the introduction of fixed drug combination of ART. And he is also a member of the Pakistan Medical and Research Council for surveillance of antimicrobial resistance in Pakistan. His work on COVID-19, I believe everybody is familiar with it. And he's also leading multiple trials on COVID-19 within the country. We hope we can learn from him today. Thank you, Dr. Faisal, for joining us. We have Mehreen Arshad with us today. Dr. Mehreen Arshad is an attending physician of infectious disease at the Anne and Robert H. Lurie Children's Hospital in Chicago, and she's an assistant professor of pediatric infectious disease at the Northwestern University School of Medicine in the US. Mehreen has joined us for multiple infectious disease pediatric related webinars. She focuses majorly in pediatric and infectious diseases. She's been leading a lot of research in this particular area, and we'll be asking her today if this particular variant does or does not have more transmissibility in children, whether we need to focus more on protecting our neonates and our under 5 children population or not. So thank you, Dr. Mehreen, for joining us today. Last but not the least, we have Dr. Daisy Iligan from Philippines. Dr. Daisy is a celebrity figure in Philippines. She also does not need any introduction. She comes on multiple media shows. She talks a lot about infectious disease, COVID-19. She's one of the leading physicians in Philippines and infectious diseases. Dr. Daisy works as the head of Dr. Daisy is the head of the TB committee at the UST Hospital. She's the chair of infectious disease prevention control at the Diliman Doctors Hospital. She also teaches physiology and medicine at the University of Santo Thomas. So thank you, Dr. Daisy, for being able to join us in today. So I'm hoping that today's discussion is going to shed a lot of light on the myths and misconceptions that we have around the new UK COVID-19 variant. There are certain information that we read in media that are not in fact true. A panel of experts today will be explaining to us what is true and what is not true. So I'm just going to stop here, and I'm going to ask Dr. Zara to please come on screen and walk us. Before we start this conversation, we need to understand, what does the SARS-CoV virus look like? Why are mutations important or not important? And what impact do they have on our immunity and our antibody response? So Dr. Zara, could you please just join in, and I'll hand off the mic to you. Thank you.
Thank you, Wajiha, for that introduction. It's a pleasure to be on this panel and share our experience on working with SARS-CoV in Pakistan. I'll talk a little bit today from the perspective of the virus, the way it's been known to change from the perspective of both diagnostics and the current scenario where people are concerned about how the virus is changing and in terms of what it could mean for generating immunity in the host. So first of all, I have a figure on the screen just to highlight some of the main components of the virus that we keep hearing about. You know, all viruses have an envelope which encases the virus nucleic acid. And so envelope is the E protein, there's the M, which is a membrane protein, and then there's S, which is a spike protein. And this word spike has become very important in the context of SARS-CoV because the SARS-CoV spike protein is thought to help it bind to the cell that it enters. And the concerns about generating changes at the level of the genome can be related to any of these proteins, okay? So in terms of perspective of the virus, there are both structural and non-structural proteins. The structural proteins I showed you on the previous slide, so there's S, there's E, there's M, and changes can occur. So the first important change that people noticed in the spike protein was related to a change in a mutation which was at the 614 residue. And that was observed very soon after SARS-CoV was identified. And essentially, there's a whole field of pathogen epidemiology or genomic epidemiology, which is related to trying to understand how the virus changes at the level of the nucleotides. So from January in 2020 to now, we have got a lot of information about genomes of SARS-CoV strains that have been identified across the globe. And it's the first time that so much data has been shared, you know, tens of thousands of sequences available globally. And the virus does change, as all viruses. And essentially, the virus changes have been linked to the development of different lineages or families across the globe. And the main families are the A and B, and the diversification of the B lineages have been found to be the most common. So there's a B, which is the largest lineage, then there's a B.1. And so a little bit about what we have seen in Pakistan, because at the Arshad University, we've had the opportunity to look at some virus strains from the beginning of the pandemic in March. And of course, we've looked at a subset of strains, but and the nomenclature becomes a bit complicated because people will have A and B lineages, but then there's type 1 to type 6 lineages, then the gissade clades are L, V, and S. But to summarize that, the predominant strain globally are related to the G lineage. And in Pakistan, mostly we have seen the G lineage and the GH lineage. So initially we saw what came out of Wuhan was the S lineage, and that split from S to L, and then L became V and G. And now most global strains are the G lineage, and predominantly that is what we are finding in Pakistan. So in terms of diagnostics, does the variety in the lineages make a difference? The answer to that is really no, because the diagnostics for SARS-CoV have been designed to be fairly robust. And the PCR-based testing, which is based on identification on genetic changes, is for more than one gene. So there's been PCRs to the N gene and the ORF gene or the E gene, and multiple PCR targets are used. So changes in the nucleotide sequences does not make a difference in identification of the virus. So talking a little bit about the Pakistani strains further, what we did was we have mapped, and this is some very initial work where we mapped a number of strains in terms of the global lineages. And this is how one tries to get a perspective of what the relatedness is with global strains compared to what we are seeing now. So we found a whole variety of countries that our strains were matching with. And these were strains from the US, from the UAE. We found the strains to be similar to those found in Iran, Iraq. So there's a big diversity. And that's not so surprising, because some of the strains came from people who were travelers, and then some of the strains were local strains. And as expected, there's a lot of commonality in the strains, and we will learn more as we go along and we are able to get more data on Pakistani strains. That's very important, because currently the data we have is mostly on Karachi strains. But Pakistan is a big country, and there may be variety across the country as well as to what strains are predominant in different regions. So that's an important thing to learn as we do more and more pathogen genomics for SARS-CoV. So the question for today was to think about the context in which changes in the new variant or the variant under investigation, mostly identified in the UK, has been, and what impact does it have. So about 23 mutations have been identified to occur in this variant, and a number of them cause changes in amino acid structures. Now, the one that has caught the most attention is a variation in the spike protein. And the spike, again, just to remind you, is the protein on the virus that helps attach to a cell and enter the cell. And there's a change that is thought to make that entry modified from the original virus, and that is possibly linked to increased transmission. Now, whether or not it is in fact more transmissible, only clinical studies will show. But from a genomics perspective, I would just like to say that if you notice, the variations in this strain occur in different parts of the SARS-CoV genome. So it's not just spike, it's in the ORF, which is the non-coding region, and it's in the nucleocapsid protein as well. So what does it mean to have these changes in the virus in the context of generating immunity? For that, we need to have a think about what immunity means to the host. So both bacterial, fungal, and viral pathogens are seen by the host by different arms of both innate and adaptive immune responses. So your innate are, just to remind you, are things like macrophages and NK cells and cells that are like the first line defenders. And then you have adaptive, slower responses generated by B cells and by T cells. And then these tend to be really specific So specific responses generated by exposure to the virus or by immunity from developed through a vaccine would be driven by B and T cell responses. And the question is your B cell response going to work, and is your T cell response going to work if the virus changes? So, of course, nothing will answer that better than evidence-based data. But theoretically speaking, one can, it's important to keep in mind that the B cell will make antibodies to all of the virus, all parts of it. And the T cell also will recognize all the different structural parts of the virus that are exposed. So it would be the S protein, the E, the M. And in fact, this is what's been shown in different studies because as recent publications are addressing these points, there are studies that have shown that by exploring T cell responses to different parts of SARS-CoV, it's been demonstrated that both CD4 and CD8 T cell responses have been seen to different components of the SARS-CoV, like the ORF1, the spike glycoprotein, the nucleocapsid protein. And then we mustn't forget about antibodies, because when we first started to learn about SARS-CoV immunity, it was mostly antibody responses that people focused on, which were driven by B cells. And the antibodies also part of the way that infection and disease and recovery from disease are monitored. So the antibody responses are known to occur also against more than just the spike protein. They're known to occur against the nucleocapsid protein. So in summary, and I'm opening it up to my other speakers, but the variety in diversity of the SARS-CoV virus occurs in different components of the genome. And because the immune responses are also to different components of it, both the structural and non-structural protein, therefore, having some changes only in one part of the protein, like the spike protein, may change the biology of the virus in a sense that it may perhaps make it bind more effectively or enter or become more transmissible, but it should not take away from the fact that the human host will be able to mount an immune response against the virus. Thank you.
Thank you, Dr. Zara, for summing this up beautifully. I think you've given us kind of some hope that everything is not lost as of yet. There are multiple, we have humoral immunity, we have T cell immunity, and even if there is a slight modification in the and mutations happening in the virus, it's not lost completely as of yet. We do have hope that immunity will kick in, vaccines hopefully will work, because just a structural change in the spike protein will not just lead to a complete loss of immunity. So thank you, Dr. Zara. I'm going to now invite Dr. Adeel Bhatt, who's going to walk us through the different kinds of vaccines available, live attenuated, the conjugated, the RNA ones, and let's wait to hear from his side. Dr. Adeel, the mic is over to you. You're muted.
Mute myself. I was trying to unmute myself, but now it was giving me a little trouble. Let me try to share my screen with you.
Sure. So if you
let me know when you
where Dr. Zara kind of left off, and we'll try and piece together how immunity, and then what natural immunity, and then what vaccines do for acquired immunity.
So let me know when you can see my screen, because I'm having a little difficulty in sharing it.
Are you doing the screen share option?
Yes.
Oh, it's there.
It's there? Okay. All right. So first of all, thank you, Wajiha, and the Getz team for inviting us to share some of our findings and the knowledge that we've gained over the last several years or so. I had prepared a more technical talk, but in the middle of this talk, when Dr. Zara started speaking, I changed some of the format into a more easily understandable format. I will quickly go over the type of vaccines that are available, the way we create vaccines and give it to humans. What are the vaccines which are available for SARS-CoV in various clinical trial stages? And then I'll put together a set of very simple questions which are on everyone's mind, and I'll try to answer them based on the available data that we have. So this slide shows some of the ways we make vaccines and administer them to humans against various pathogens. So there are about 6 types of vaccines, 5 of which are shown in this slide. You have DNA and RNA-based vaccines. These are essentially genetic component of a pathogen, of either a virus or even a bacteria for that matter, that can be inserted into humans, and that elicits an immune response from the humans by producing a protein that is unique to that virus. Now, I want to be clear that no DNA vaccine has been approved yet. No RNA vaccine has been fully approved yet either, but at least 2 RNA-based vaccines for SARS-CoV-2 have received emergency use authorization in multiple countries, and we will talk about that. Then, of course, you can inject inactivated virus or a subunit or a subparticle of the virus, which is a unique protein that the human body will recognize and produce antibodies to. You have vector-based vaccines. You insert the genetic material or some part of the pathogen into a vector, often an adenovirus-type vector, and that is injected to elicit an immune response. And then you have live attenuated vaccines, where you take a pathogen, you inactivate that or attenuate its pathogenicity by taking off the key components, pathogenic components, and then inject that. In terms of SARS-CoV-2, we have 4 vaccines which are in advanced stages or have received emergency use authorization. Then you have several others which are in various clinical trial stages yet. So, of course, the greatest interest has been generated by the Pfizer vaccine, the Pfizer or BioNTech vaccine, which received emergency use authorization in the US, and several countries have started administering that. This is the vaccine that I have received the first dose myself as well, so I'll be happy to share my personal experience with you. The other RNA-based vaccine, mRNA-based vaccine, is the Mrna vaccine, which was jointly created with the NIH, NIAID, and Moderna. Then there is the Oxford AstraZeneca vaccine, Novavax vaccine, and in Pakistan, I know there is a lot of interest in the Chinese vaccines because they are in clinical trials in Pakistan. We will briefly touch about that. And there's Sputnik vaccine, which is the Russian vaccine. We hear about that a lot in press releases and media, but I haven't seen hard data on that so far. This is the stage of vaccine development a couple of weeks ago, and this definitely would have changed by now because, for example, approved vaccines, the emergency use authorization has increased to 3 because only yesterday or the day before, the Oxford vaccine was approved in the UK. There are more than 50 vaccines in phase 1 trials right now, several in a couple of dozen in phase 2, and nearly 24 in phase 3 clinical trials, which will be advancing, and we'll start seeing data very quickly. Since the first 2 vaccines to be approved were mRNA-based vaccines, so a quick slide about what does this mean? How does this work? So I want to be very clear. No infectious virus or intact virus, whether live, attenuated, or dead, is inserted into the human body in these vaccines. They take, scientists take a part of the genetic code of the virus. They attach other nucleotides to both ends to make it stable, for it to be able to enter the cells, and then uncoat itself once inside the human cell. But the green part that you see on the slide is the genetic code or the RNA. Once inside the human cell, it uncoats and translates and expresses the protein that it is supposed to produce. In this case, in the case of SARS-CoV-2, it's a spike protein or parts of the spike protein. And this spike protein is then extruded, and that is what elicits the human immune response. Now, here we'll start off with a set of simple questions that I keep getting on. So now that we have a few vaccines, the most common question I get asked both by my physician colleagues as well as lay people is, which one should I take? And my answer to that is whichever you can get soon among the approved ones. At this point, we don't see a huge difference in the efficacy rates of various vaccines to recommend one over the other. There are certain pros and cons to each one, and I'll share a little bit of data about the 3 vaccines that we have started using. So the first one is the BNT162b2. This is the Pfizer-BioNTech vaccine. So a lot of interest has been generated and was the first one to get widespread use in the US and in many other countries. So in summary, I have technical slides if someone wants to go into the details of the trial as well, but I'll summarize the data. They took about 40,000+ participants and divided them into the vaccine group and the placebo group, and found out that in the placebo group, there were 162 cases of SARS-CoV-2 disease, symptomatic COVID-19 disease, and only 8 or 9 cases in the vaccine group. So this translates into a vaccine efficacy of about 95% or so. In terms of severe COVID disease, there was none in the vaccine group, and there were 13 in the placebo group. But actually, this is for the other vaccine. Even in the Pfizer vaccine, even in the Pfizer-BioNTech vaccine, severe COVID disease and deaths were not really seen in the vaccine group. In the other study, which is the Mrna study, there were about 185 cases of disease, symptomatic COVID-19 disease in the placebo group, and 11 cases of disease in the vaccine group, again translating into an efficacy rate of about 94.1% or so. Now, the third one is the Shadok AstraZeneca vaccine. This is the Oxford vaccine. Again, this trial is a little more complicated to interpret, but does show a high level of efficacy. And that depends on the specific sequence that they use, low dose, standard dose in various sequences. And overall efficacy of this vaccine was a little over 70% or so, but there were certain subgroups with the best efficacy rate of about 90 to 92% or so. So again, quite good efficacy, not quite reaching the level of the other 2 mRNA vaccines. Now, this is not an mRNA vaccine. This is based on this is a vector-based vaccine. So which one should I take? I said whichever you can get soon. I prefer the mRNA-based vaccines right now because they show a better efficacy overall at this point in time. And we have had a few million people inoculated worldwide to give us some sort of assurance that these are relatively safe to use. Another question I get asked all the time, should I take 2 or 3 vaccines? For example, if I've taken the Pfizer, should I also take the Moderna, or should I also take the AstraZeneca vaccine? And at this point, the answer to that in our mind is no. The incremental benefit of getting multiple vaccines would be minimal, but adverse events would likely be additive. Also, we do not know how this will affect immunity if you give 1 or 2 or 3 vaccines, or if you mix and match vaccines. Now, I know the UK approved this mixing and matching in certain instances when the same vaccine is not available, but Dr. Fauci and NIAID and NIH in the US, CDC in the US cautioned against that. We do not know enough about this to recommend this mixing and matching at this point in time. And we do not recommend the combination. You can ask, if vaccinated, can we travel and stop all these precautions like wearing masks and physical distancing? And our answer to that is no, not initially. And this is a point of contention for a lot of people, saying that if I'm getting the vaccine, what is the point of getting a vaccine if I can't go back to normal? And we give caution to that we need to wait till large proportion of the population is immune or develops, or populations develop herd immunity so that the transmission can be stopped. While they're highly efficacious, the efficacy is not 100%. Even with 95% efficacy, there's some people who will still get the infection and disease. If I've already had COVID infection, should I still take the vaccine? And if it is available, then our answer to that is yes. And the reason for that is that immune responses after natural infection are variable, and they're also dependent on the severity of symptoms. In other words, if you have had milder disease, it is likely, some data show that you may have lower level of antibodies and hence lower protection against subsequent infection, while vaccination produces more reliable and high levels of humoral and cellular immune response. So it is more likely that you'll get a good immunity with with the vaccines that we have. We have had a lot of questions about allergies, so that I have had allergies or I have a history of allergies, can I still take it? Unless you have had an allergic reaction to a specific ingredient, or you've had an anaphylactic shock to any external stimulus, the answer is yes, you can still take it. And we do monitor people for a little while after the vaccine has been given. So having allergies is not a contraindication. You just need to be a little more careful and monitor. Only if you've had anaphylaxis, you need to be very, very careful and administer and monitor the patient in a good healthcare setting. We keep getting asked if I'm immune suppressed for any reason, or I'm taking immune suppressive medicine, can I still take the vaccine? All the current trials excluded patients with immune deficiencies and immune suppression. However, since there is no live or even attenuated virus in the mRNA vaccines, it's generally safe for people to take even in immune suppressive conditions. However, we are not certain how the body's immune response to that would be. But in terms of safety, I don't think we have seen any major signals at this point in time. And the expert consensus is that yes, you can still take it. There are no data for children or pregnant women. The trials in children under the age of 18 have started. The first set of trials is for children between 12 and 18, while pregnant women will be enrolled in trials very soon, or some have already started actually. So at this point, we have no data. However, again, the expert consensus, at least for pregnant women, is that it may be okay to give it to them, but that has to be discussed on an individualized basis. Question is, how long does the protection last once I take the vaccine? And we do not know the answer to this completely or fully, because we have only had about 3 to 4 months of follow-up after the clinical trials. But it is likely to last at least 4 to 6 months, and possibly longer, up to a year or so. This is based on early phase data from phase 1 clinical trials and immunogenicity trials, also based on data from those who have been naturally infected, and we followed the antibody titers for a while. People are worried that can I get COVID from the vaccine? And the answer to that is no, you cannot, because there is no virus in the vaccine, live or dead or attenuated. So it is not possible for you to get infection from the vaccine. Will I need the vaccine every year or so? And again, we do not know the answer to that yet. As I mentioned earlier, immunity is likely to last for at least several months. So hopefully by that time, the global pandemic will be over, and we hopefully will not need it every year, but we'll have to wait and see. Can I transmit COVID even after I've been vaccinated? And the short answer to that is it is possible, although it is quite unlikely. That is why we still say that you must follow all precautions till immunity levels in the community are high enough to achieve herd immunity level. Also, there is a certain window period or lag period particularly after the first dose, where you may harbor the virus initially and may still transmit the virus before the vaccine efficacy has kicked in. What if I miss the second dose? And again, in the UK over the weekend, there have been some mixed signals about this. They have said that you can wait for the second dose for a long period of time. We do not know what a fair window would be, but if you have missed it, we recommend taking the second dose as soon as possible. And if you have missed it, there's no need to get the first dose again. You can just take the second dose and go on with it. A question gets asked that isn't natural immunity safer than the vaccine? Should I just get natural infection rather than going for a vaccine? And again, the short answer to that is no, it's not. Antibody levels after natural infection are variable and not universal. The level of neutralizing antibody is unclear. Vaccination, on the other hand, provides reliable higher level of protective antibodies. Also, there is little or no chance of getting COVID-19 from the vaccine, while with natural infection, there is a risk of getting severe disease and dying from it. The case fatality rate of about 2.5% to 3% at this point in time. If you're young, healthy, and at low risk, why should I take the vaccine and take that chance of side effects? Again, I would say that the overall risk-benefit ratio heavily favors taking the vaccine. Will these mRNA vaccines alter my genes, my DNA? Will I develop a tail? Will I grow a tail or horns or some other weird things. A lot of memes are going on social media. The answer to that is no, you will not, because mRNA does not enter the nucleus and does not integrate into the host genome. After it produces the protein that it is supposed to produce, it disintegrates. So there are no chances of it altering your genes or your DNA. So here's the big question which we're discussing today. How will the recently reported mutations affect response to vaccination? Now, as Dr. Zara pointed out, that the new variant, the V1.1.17, has 17 to 23 mutations, some of which are on the spike protein and the receptor binding domain and so forth. However, it does not seem at this point that this will alter our immune response to the current vaccines. There are no data, no hard data, but our expectation is that so far, at least with these number of mutations, we are not seeing any alteration in your immune response to vaccination. I will stop here and will be happy to answer any questions when the session comes up.
Okay, so Adeel, while you were talking, we did receive a lot of questions, so I'll keep it for the QA section. So thank you very much for walking us through all the different categories of vaccines. I believe, Dr. Faisal, you will be taking over now, and what I want you to point out towards is two things, basically. We expect you to please talk about the status of clinical trials in Pakistan as they're happening, the Cancino bio and the Sinovac one. And do they have any side effects? Should people come and get enrolled for it? Also, if because you are in talks with the government for getting various other vaccines, is there any hope for getting vaccines in the first quarter of 2021 for the general population of Pakistan or not? And the third part that I really want you to emphasize is on the fact that because of the change in mutation, will the vaccines, what are the kind of vaccines that might fail because of the change in the spike protein? The category of vaccines, you don't have to name them. You can.
All right. Thank you, Wajiha. So thank you very much. So I actually do have 3 slides. I promised no slides, but I made some while we were going, while the other talks were going on. And I'll probably take these 3 questions in reverse order. So first of all, will the vaccines be affected by these mutations, or will the new variant be affected by the mutations? And then we'll go on to the vaccine trials in Pakistan, and then finally when the vaccine will come over here. So I'm just going to quickly share my slide. I only have 3. So that would be right over.
They're all very powerful questions, and we have a lot, relying on you. It's in a sequence. Just build up on what Zara and Adeel have been talking about.
So can you see my slides?
Yes, we can. Please go ahead.
All right, so I'm just going to pick up from where we left off, and that's the reason why I'm going to start over here, because it's fresh in everybody's minds, and exactly what Adeel had mentioned. Just to revise again, we have these 6 types of vaccines: the inactivated virus, where you inactivate the virus; the live attenuated, where you change the virus a little bit, so it can't replicate, but it's still the SARS-CoV-2 virus. You have the recombinant viral vectors, where you put the gene of the spike protein in under different virus, so you start making the spike protein. The virus-like particles, where you have the spike protein studded on a membrane. You have the RNA, DNA, and then you have the protein subunit. So, and the reason to bring this up again is to focus everybody's attention on the fact that pretty much most of our vaccines are focusing on the spike protein, and that's where one of the mutations or 2 of the mutations have occurred, and that's where the concern is that will these vaccines now work? And the goal for the vaccine for our lay public essentially is that we fool the body that they're getting infected by showing them the spike in various forms. It could be as a whole virus or just the DNA. And then you develop antibodies against the spike, and we know these antibodies against spike will help neutralize the virus. But the important thing, and this is now building upon what Dr. Zara had mentioned, is that the spike protein, which is here out studied, really is not just one. It's like a huge protein with many, many parts on it. It has an S1, this this round walla part, which binds to the receptor, which binds to the cell. And it also has this little stem over here, which is the S2, which is used in fusion. So after it binds, it has to fuse onto it. And the antibodies you make against spike, even though we just say spike antibodies, you don't make one antibody against spike. Spike is a huge protein. It has many epitopes. studies show 66 places where the antibodies can bind. And depending where you develop antibodies, for example, if you develop it to the receptor binding domain up here, you'll block this interaction right here. If you develop it against this round thing over here, you won't have a conformational change. If you develop antibodies against the stem, you won't have fusion. But the end result of all of these is exactly the same, that the virus will not be able to enter the cell, and this would be an effective antibody. So the short answer to your question, really, Wajiha, is that these vaccines will almost certainly work against these new variants. And in fact, one of the mutations, this variant, the 501 Y mutation, has already been looked at to see if antibodies if the antibodies produced against SARS-CoV-2 are they effective against mutants with this mutation, and it was found that it is, and this was in a hamster model. So we know that it'll work. The reason why there's concern is that we know that the virus mutates, all viruses mutate, but the concern is that with growing mutations, we may run in trouble in the future, but currently not right now. And a good example of this is right here. You can recognize this face regardless if they're wearing glasses or not, and your antibodies can recognize a spike regardless if there's a little change in it or not. So that's the answer to your first question. I'm going to stop sharing and come back over here, and I'm so sorry. I'm going to change my screen.
That's the best. Okay, guys and girls, just summarizing, vaccines are still going to work. It's a media hype, and Faisal has very beautifully explained that the face remains the same, even if you just put glasses to it. So the vaccines are not that stupid. They're going to understand particular bits and pieces of the virus.
Exactly. So it's almost like if I have a big pimple on my face, you still know it's me, right? So to answer your second question then, which is what's the status of the vaccine trials in Pakistan? So currently we actually have 2 vaccine trials ongoing. There is one which is a phase 1 vaccine trial, and that's a very small trial, it's almost finished. But the more important is the Cancino vaccine trial. Now the Cancino vaccine is an Adenovirus 5 vaccine. So what we've done is that we've taken the spike protein, DNA, or RNA, we put this inside a different virus, an adenovirus virus. This is a very harmless virus. This virus has also been modified so it can't replicate. So once somebody's injected with it, we can't get this infection. And then you start making the spike protein after the injection. So Pakistan is one of the sites for this in the country, in the world. We have been given a target to vaccinate at least 18,000. We're now up to about 15,500 people who've been vaccinated. In fact, as we speak, surprisingly, I just got the message for today. So we're up to 15,698 people who have now been enrolled in this trial. So we're very, very close to the end of 18,000 folks. Remember, there's a randomized control trial, and blinded, which means that half the people would have gotten the vaccine and not get the vaccine. And then we'll see how many people eventually down the line develop COVID-19. We haven't seen any major side effects. In our side, the major side effects we've seen really is body aches and pain at the injection site, which is very common. The other side effect that I've seen in our side is a little bit of rash. And I've seen this in about 3 people. So that's 3 out of, now we vaccinated close to 2,500 people. So the rate is really low of rash. So not very, very common. No major allergic side effects that we've seen in the others. So it's a fairly safe vaccine. We do not have enough to do the interim analysis as yet. In other words, we don't know if it works or not, but we will soon, based on the way the number rates are going in the country. And then finally, your first question is, when will we get the vaccine in Pakistan? And this is a much more tougher question, actually. We probably will get the vaccine probably by first quarter, but this is going to be a limited supply only for the frontline workers, for people over 65. Which vaccine we get is something that we can't say right now. You may have seen the news of one of the vaccines that was already bought, but it had to get DRAP approval. So, and that I know talks are underway currently to get DRAP approval for those. And DRAP for people outside the country is the Drug Regulatory Authority, sort of like the FDA of Pakistan. So until that gets through, we really can't do the thing. So that's the 3 questions you asked, and I'm going to hand over to the next speaker, if it's okay with Dr. Wajiha.
Okay. Just one last question while you were speaking. So, how long will is the Cancino bioimmunity supposed to stay? Like, are we supposed to get boosters every 6 months, every 3 months? Any idea? Because this is an old technique. Adenoviruses have been around, adenovirus vaccines have been around for a long time. Do they have a long-term immunogenicity?
So the adenovirus vaccines really do have a much more robust immunogenicity, especially the Ad5 vector, which is why for this particular vaccine, it's actually there's no boosters, a single dose only, which is one of the benefits of this. Similar to the Johnson vaccine, which only also has a single dose, while the other ones you require 2 doses. How long the vaccines will last for is sort of a tougher question. The immunity. We've had adenovirus vector vaccines around, lots of V's in that sentence. But having said this, I would say exactly what Dr. Adeel said, is that we think it's probably going to be a year or longer before. And, once the phase 1 and 2 trials results come, we'll probably get a better idea.
Right. So I do have a lot of questions, but I'm going to leave it for the QA session now, handing over to Dr. Mehreen. Another very important question that we get asked is that there has been a lot of news around, especially from media, that this new strain B.1.1.7 is more transmissible in young school students, not in adolescents, but in children. Is that true? If that is true, what is the reason behind it, and what are the kind of impacts that we'll see in overall society measures? Will schools remain closed for another quarter or so? Again, I know that you might not have answer to all questions. It's still fairly new, but if you could just talk about the data that we do have available at the moment. Thank you very much.
Thank you so much, Dr. Wajiha. So, Dr. Faisal, I was going to try and share my screen. All right, thank you so much. So thank you so much for the invitation to talk today. I think this is a very important topic that has come up recently, and I will say that there really isn't a lot of information and data out there. So what I'm presenting today is mostly sort of what is in the media and what is being put out in really small reports. All right. So if you go to the next slide, and the next slide. So what I really wanted to start off today was talk a little bit about what we already know about children and COVID-19, just as sort of an overview of where we are in the pediatric infectious disease world. So COVID-19, and this is sort of summary of lots of other studies and papers that are out there. So COVID-19 is reported, but it is much less frequent in children than in adults. If you sort of look at it on a global scale, children and adolescents represent about 8% of the reported COVID-19 cases. Most of them, the pediatric patients have mild and asymptomatic infections, and they are, and they're actually fairly likely to be underreported as well, because a lot of kids, because they have this mild infection or are asymptomatic, will not present to a healthcare system and get tested. Children, as I mentioned, have much milder disease than adults. However, children who have underlying conditions, and these could include anything from structural lung and heart disease to those who are immunocompromised, those who are obese, for example, are at higher risk for serious illnesses. And we have, in the US, have had more than 100 deaths in the pediatric population from COVID-19. Rarely, a few may also develop what is called multi-system inflammatory syndrome in children, or MIS-C. This is a disease that is essentially a post-inflammation, a post-COVID-19 inflammatory syndrome, and it usually occurs about 2 to 4 weeks after the onset of COVID-19. Most of these patients recover, but there have been deaths from MIS-C reported as well, both in the US and in other parts of the world as well. The role of children in transmission is not fully understood. We do know that children of all ages can be infected and they can spread the virus to others. But studies do show that there's less spread among children under 10 years of age than in older children. And when infections do occur, they're more likely to occur in teenagers than in younger children. And older children also seem to be more susceptible to infection and to transmit more often than younger children. All right, next slide. All right, so this is one of the largest studies that is currently out there. This is the study that was put out by South Korea, and they had thousands of people that they had done excellent contact tracing on. And so what they did was they traced all the contacts of the person who initially presented to the healthcare system and was tested positive. And what this slide is really showing is that if you look at the children who are between 10 and 19 years old, and if you look at the household contacts, about 18.6% of the household contacts were positive, which is the highest amongst all other age groups. And if you look at the 0 to 9 age group, 5.3% their household contacts were positive for COVID-19. And so clearly, the 10 to 19 year age group, there is a higher risk of transmission to family members or people who basically live close to that child. Now, they did not see the same rates of infection to non-household household members, but again, this was sort of in the setting when schools were closed and children were not sort of really getting out of the house. Next slide, please. So in summary, if you look at all the school data that is out there, there have been a few outbreaks that have been reported in schools since early 2020. A lot of the initial data that we had was in the setting of school closure anyway, and a lot of children were staying at home. So it's not very reliable data. We have had more data come in since schools have opened, and what that really shows is that again, most of the infections of COVID-19 cases that have been reported in children, the infection was initially acquired at home. More outbreaks have been reported in secondary and higher schools than in primary and elementary schools. Again, this sort of goes along with the data that I showed before, that the 10 to 19 age groups are more likely to spread it to their peers within a school. And in school outbreaks, it was more likely that the virus was introduced by adult personnel, and so transmission has happened between staff to staff, and this is something that we've seen even in our pediatric hospitals, that it is much more common to see an adult to adult transmission happen in the children's hospital rather than the pediatric patient or a student to adult transmission. And student to student spread did occur in schools, but it was much more rare and tended to happen in the older, in the secondary and higher schools. An early modeling study suggested that closing schools reduced community transmission, but less than other social distancing interventions. So if you close schools but don't close restaurants and bars and cinemas, for example, and malls, that the impact on society and the impact of COVID cases in the country were less likely to be affected. Next slide. All right, so the bigger question is, with this new variant, are children getting more infected with SARS-CoV-2? And so there's very, very little data out there right now, but I did want to show a couple of statistics that have come up in the last few weeks. So this is data. I actually could not find age-related data from Pakistan, but this is U.S. data, and what that really shows is that over sort of November, we started to see many more cases of COVID-19 in children. And if you look at the last 3 weeks of December, we've really had, this is the number of pediatric cases has almost doubled since, just a few weeks before that. And then the next slide. And this is really the data that has sort of generated a lot of discussion and a lot of, concern among school authorities as well as parents and pediatricians. This is data from UK, and this is basically the data from the first week of December. And that showed, if you look at the very first graph that's on the left-hand side, this looks at the estimated percent of the total population that tested positive for COVID-19 from nose and throat swabs. And so what you'll see is a school year age group from 7 to 11, that age group had 1.9, so 1.9% of all children in the UK were positive for COVID, and their percentage is higher than any other age group. Now, hospital admissions and deaths continue to be higher for 85 and over, as expected. But because this population is this population of children, if they are getting more infected, and if this data is true and persists over time and is related to the new variant of COVID-19, that has a huge impact on how we deal with school closure and school opening and the mitigation measures that we take as a country and as a society. The next slide, please. Right. So how should we interpret this data? Now, one thing I will say, being in the U.S., the U.S. pediatric cases, again, there have been cases of the new variant, reported cases of the new variant in the U.S., but at the moment, we don't believe that it is as widespread as in the UK, for example. And we do know that recently we had the Thanksgiving holidays in the US, and despite a lot of recommendations to not travel and not meet, there was a lot of sort of travel and lots of family meetings and things like that, and perhaps that led to an increase in the pediatric cases in the US. In the UK, schools have largely been open, and I will say, both from talking to people that I know who are in the UK as well as reading some of the media and the news reports that are coming out of UK, they really haven't been very good with strict mask and social distancing protocols. So I kind of feel that before we sort of go down the route of children being a major reservoir of this virus, or being a major source of spread of this virus, we really need to make sure that we're doing the basics, which is masking, social distancing in school, making sure that children who are sick are not coming. So I think all of those factors really need to be accounted for, and we need to make sure that we're doing all those protocols in school. What we also do know is, because children are more likely to be asymptomatic or very mildly symptomatic, positive cases in children are likely being underreported. So that 1.9% of children who are positive at the 7 to 11, that may actually be an underestimation of the true number of children who are infected with COVID-19. Because as we've talked about before during this session, it does not seem that the new variant is more pathogenic, so it doesn't cause more severe disease, but it's just much better at being transmitted. However, if children are more susceptible, and even if they're not likely to get more sick themselves, they will school opening and things like that will need to be reevaluated. And again, as I said, there are children that get very sick and have died from COVID-19, both acute COVID-19 as well as the multi-system inflammatory condition. And if you have a higher number of children getting infected, then the number of children who die and who get more severely sick from this virus will also increase. So obviously this is very concerning. And the next slide, I just quickly wanted to mention. So I just quickly wanted to mention multi-system inflammatory condition, because this is something that we are seeing much more frequently in the U.S. In my institution, we are seeing about 6 to 7 cases per week. We used to see 1 to 2 cases per month till November. So this is sort of been a significant increase, and it's pretty concerning. And the kids who present with MIS-C are very sick. About 70% of the children in our institution require ICU care because they present in shock. And so these children present with fever, they present with cardiac dysfunction. About 50% of them have abnormal echoes. They have heart arrhythmias. They have respiratory manifestations. Their GI symptoms are very, very common, and so a lot of them will have fever and very intense abdominal pain. A significant number of them have been initially diagnosed with appendicitis and then diagnosed with MIS-C. So just something to sort of keep in mind as the number of pediatric infections with COVID increase, there will be a higher number of patients who present with MIS-C as well. And the other thing I will say is, unlike severe COVID that tends to occur in children who have more who are sort of have underlying disease. MIS-C tends to occur in children who are otherwise completely healthy. All the children that I have taken care of with MIS-C had very mild disease. They were completely healthy prior to presenting with MIS-C, and then got very sick very fast and ended up in the ICU. We have not had any deaths in our hospital, but it is pretty scary when these children present to our ER. We'll stop there. Thank you.
Great. So thank you so much, Mehreen. Amazing presentation. Got lots and lots of questions. We have one last panelist, and then I'll start asking questions individually. So thank you, Mehreen. Now we have with us Dr. Daisy, who's going to elaborate the status of COVID-19 in Philippines. She's going to talk about the wave 2. She's going to talk about measures, the vaccine trials, etc., and her own particular knowledge as is being seen in Philippines. So thank you, Dr. Daisy, over to you.
Hi, yes. Good evening to everyone. No, I would like to thank the Getz Pharma for inviting me. So it's a different topic as compared to the other speakers. Now I am going to talk about the Philippine experience and how the government is dealing with the pandemic. Later on, I will just share one slide regarding the current status of the Philippine COVID-19 pandemic. So the Philippines' first case of COVID-19 was documented last January 30, 2020, who was a Chinese national. And the first local transmission of COVID-19 was documented last March 2020. So one of the greatest challenges that we faced during the first few months of pandemic is the lack of facilities, including the centers for RT-PCR for SARS-CoV-2. Can you imagine, just initially, we only relied to one testing center in the Philippines, the Research Institute of Tropical Medicine. However, the government was able to expand and try to meet the demands of the increasing cases of COVID-19 by increasing the centers, both private and government-owned testing centers. So it was reflected by in the Department of Health COVID-19 case tracker that we have a sustained high number of cases from March to June due to the increased number of testing capability. Research trials, for which I am also included, the Solidarity Trial in cooperation with the private practitioners, World Health Organization, and the Philippine Society for Microbiology and Infectious Diseases, were started in search for the treatment of COVID-19. So we started the patient initially with different medications like hydroxychloroquine, and then we found out it's not working to our patients, and even the ARV medications like lopinavir, and then later on was not part anymore of the trial because of the data showed that it is not having any help in our COVID-19 patients. Many other trials are also being conducted in different hospitals. So another challenge is the lack of hospital facilities and personal protective equipments. Some government hospitals were converted to COVID-19 specialized hospitals and private hospitals, and the private hospitals were even mandated to accommodate and manage COVID-19 patients as well. So many private hospitals or individuals and even private sectors cooperated in the fight against the pandemic of COVID-19, especially regarding the shortage of the personal protective equipment. So we will just receive donations from private sectors giving us free personal protective equipment. So initially, we have shortage of different personal protective equipment, including N95 mask. But right now, no, we have different. We're able to meet the demands with COVID-19. Now, I'm going to try to share a slide from the DOH. Okay. Tracker, COVID tracker. I hope you can see the COVID tracker. So if you can see, we have a sustained number of COVID-19 cases from March to June, and we have an increased number of COVID-19 from July up to until August, and starts to go down last September 1. So the new strain of SARS-CoV-2 D614, I'll just try to stop it. The D614 was detected by the Philippine Genome Center, which is associated with higher viral load and higher rate of transmission last June 2020. So there was a surge in cases of COVID-19 last July and August that most of us frontliners called for timeout and and to re-strategize. Strict lockdown to communities with high cases of COVID-19 were again implemented. The new strain during this time could partially be contributed to the increase in cases, but the main driving force in the rise in the case was actually the failure to comply with the government's infection protocol and the more relaxed quarantine measures. There's no evidence that the mutation has impact in the disease severity. The cases started to decrease luckily last September, and we are still hoping to continue to decrease the cases in the next months to come. So I personally believe that the decrease in cases of COVID-19 can be attributed because of the cooperation of both the private and government efforts. And until now, no new UK variant of SARS-CoV-2 has been detected, and the Philippine Genome Center is working on it. So they keep on collecting the samples from the specimens of COVID-19 to detect if there is already a UK variant present in our country. It is always emphasized by our government the potential positive impact of the vaccines to the pandemic. So November last year, we have 5 pharmaceutical companies who expressed their intention to hold the phase 3 trials in the Philippines and include China's Sinovac and Clover Biopharmaceutical, Russia's Gamaleya Research Institute, Johnson & Johnson of the United States, and the UK AstraZeneca. However, just recently, AstraZeneca withdrew the bid to hold the trial in our country. So as of the moment, we don't have yet any approved COVID vaccine available in the Philippines, but the Pfizer's COVID-19 vaccine is expected to get emergency use authorization. So as of the moment, we are still on the general quarantine, community quarantine, which is equated into still stricter protocol for the establishments, not allowing children less than 15 years old, and mandatory wearing of face mask and face shield. Moreover, the government also implemented travel restrictions in countries with documented local transmission of the SARS-CoV-2 variant and implementing mandatory 14 quarantine to travelers into the Philippines, especially among cases with these cases of COVID-19 UK variant.
Oh, okay.
Yeah, that's how our government adapted to the new COVID-19 cases. But as of the moment, we are enjoying the decreasing number of cases. But I am bracing myself because of the New Year celebration. We Filipinos love celebration, so we have some gatherings, and then we expect a little bit rise in the few months to come.
Good. Okay. So thank you so much, Dr. Daisy. One of the reasons why we wanted Philippines on board was as a kind of a case study that you can control COVID with good measures. Just like Australia has also done it, New Zealand has also done it, and we Pakistanis and you guys sitting in the US, we really need to learn. So I had a very interesting question that has come up, and it's kind of funny also. And so people are saying that if Australia and New Zealand were able to control the pandemic without a vaccine, why do we need it? So who wants to go ahead and answer that question and link it with herd immunity and probably the population size of Pakistan and the US? It's open to all. Faisal, Mehreen, Dr. Adeel. Can I please have all panelists simultaneously on the screen, because we're going to start asking questions now.
Can you hear me, Mehreen? Wajiha, sorry.
Okay. Dr. Adeel, I think you want to go ahead.
Yes, I think the answer to that is just take a trip to any mall in Karachi or your neighbor India, any street out there, and you'll get the answer to that very quickly. Everyone is walking without a mask. They are on top of each other. So yes, if we had followed some of those guidelines of physical distancing, personal hygiene, like hand washing and so forth, very strictly early on, there are modeling studies that show that the mortality as well as infection rate would have been a fraction of what it is globally right now. I think we missed the ball very early on. Another good study, case study, is China. pandemic started in China, and remember, for the first several months, China was way ahead in numbers than any other country in the world. And after the first 3 or 4 months or so, now they don't even figure in the top 20. They almost arrested the numbers at like 80,000 to 90,000 range for months at end. And that was because of very strict measures that were taken into place. So that's the long and short answer to that.
Okay. It's also probably something to do with the concept that herd immunity is going to be reached when we're 70%, and we are such a huge populous country that's going to take a little amount of time for us to reach that 90% in terms of numbers, right?
So again, I think
we need a larger volume to, I mean, I don't know. That's a question.
So I think the concept of herd immunity is vastly misunderstood by the community, and I think the panel here would back me up on that. That herd immunity does not mean that you as an individual are protected from infection. Herd immunity means that a large number of population has now been infected, and now the pool of susceptible people is very small for transmission to take place. Now, if you look at the current case fatality rate, which is globally around 2.5% to 3%, higher in some other countries, but globally it's about 2.5% to 3%. For a population of 220 million people in Pakistan, if you were to get 80, 70, or 90% people infected, the number of deaths would be monumental. So you can't wait for herd immunity through natural infection. That will be disastrous for any country the size of the population. So I think that needs to be really hammered home, that with an infection with a case fatality rate of what we see with SARS-CoV-2, herd immunity from natural infection is devastating.
Yeah, I mean, I'll just,
anyone?
I'll just add, I think in the US is a perfect example of this, because, you know, the COVID deniers over here as well, and the vaccine deniers over here have talked about herd immunity as being a way forward a lot in this country. But if you look at the statistics, we have had 346,000 deaths from COVID. And if you look at the seroprevalence of COVID antibodies, it's only about 20% at best. And so in order to get to that 70, 80% herd immunity, it's gonna be devastating, you know, and we, right now in the US, we have one in 1,000 people living in the US have died from COVID. So there is no way that we are going to be able to get to a level of seroprevalence that will be effective in providing that herd immunity without having millions and millions of deaths.
And then the other thing to add on to this, Wajiha, is that the concept of herd immunity, as we understand it, is also not correct, in that even if we develop herd immunity, the infection is going to go away. No infection has ever disappeared because of herd immunity in the world. And we always keep forgetting about reinfections, because we're already seeing a number of reinfections. Adeel had a big tip on reinfections also, which essentially means, you know, that I don't think herd immunity is the way out of it, regardless, even if we get this many people infected.
Can I just add something about, just to add to the spread of infection? There in Pakistan, we're seeing like a pandemic fatigue. So people who were masking 6 months ago are being less careful now. And that's really worrying, because in fact, it's exactly as was said before, the more infections you have, the more variety of people you will have, the more age groups you have affected. And that is spreading through the population. So everybody knows somebody who has had COVID, and it doesn't always end well. So, you know, that really needs to be reinforced.
Okay, yes, I would also to add, my personal experience. I had several patients with reinfection, so I definitely agree with Dr. Faisal. So there is a chance of reinfection, and it's going to be survival of the fittest. There will be a very, very high mortality among patients if we're going to hope for herd immunity.
Okay, so I have like over 100 questions, and I'm going to try to wrap them up in 30 minutes or so. So I'm just going to pick on the pertinent ones. So I'm going to start shooting. Just a little request. One of you can go ahead and answer the question. And if anybody has anything else to add, because otherwise I'm just going to do a rapid fire round for questions. So just shooting ahead. So if a person has had natural COVID and he or she still has antibodies in the blood, Do they need to get vaccinated? Yes or no? Anybody?
So I think I answered that question in my Q&A that yes, and the reason for that is that the antibody level or protection level from natural infection is variable, and we don't know what your antibody and protective level is. The response from vaccine is much more predictable, and the level of antibodies is more constant and much higher.
Great. If you're PCR positive, would you still be told to get a vaccine or not?
The answer is yes.
Explain
I mean, after the infection is done, yeah.
I think it depends as long as you are so within the 10, so if well, at least in my institution, if you are 10 days after the onset of symptom, and despite even if you're PCR positive, as long as you're not acutely sick with COVID, you can get the vaccine.
So if you do not have active infection, but you have remnant PCR, you can still go ahead and get the vaccine.
Yes.
Great. Again, an open thing, there have been reports of Guillain-Barré syndromes worldwide, especially Adeel, in your area. Could you just comment on it? And also for Bell's palsy.
So here's the answer, that the incidence of Bell's palsy in the general population is about 60 to 70 cases per 100,000 population per year. In other words, if you vaccinate 1 million people, about 250 people will have Bell's palsy, whether or not you were given to give them vaccine or not. So if you see one case of Bell's palsy here and there, or 10 cases or 200 cases, that does not mean it's because of the vaccine. Another pertinent example, I saw this question on the screen. I did a quick Google search. There were 6,000 dog bite cases in Pakistan in 2019. That translates to for each million people, about 200 cases of dog bites. Now, will you say that if 1 million people got vaccinated, they got dog bites, the dog bite is because of the vaccine? No, because there is a baseline rate for everything. So don't ascribe anything that you saw anecdotally to the vaccine.
Great. An open question, all the RNA vaccines that we're seeing require a cold chain storage. How do you see the success happening, especially in lower middle income countries where there is no, cold storage line for supply? Again, an open question, anybody can answer. And will the efficacy go down if it's not stored at -70, especially the Pfizer?
So the answer to the second one, if the cold storage chain is not maintained, yes, the efficacy will go down. The vaccine won't work. And this is a major issue, the cold chain, which is why even if Pfizer came to Pakistan as a vaccine strategy, it would not be the only vaccine that's there. It will only be the bigger vaccine thing. So that is a big issue, yes.
Okay.
Yeah, and the reason for that is the RNA is very unstable at normal temperature, so the RNA disintegrates relatively quickly. So that is why it has to be frozen.
Great. Anyways, I guess for the lower income countries, it's expected somewhere in Q3 of 2021. Correct me if I'm wrong, Faisal. Yeah. Okay. So let's wait and see. We might just be able to get a cold chain by then. Wishful thinking. Okay. So the next one is, since there is a new COVID strain, and I asked this from Faisal before also, which kind of viruses would be, which kind of vaccines would be affected the most? You already answered.
So I have, and just I want to make a correction. Then is that this is not a new strain. This is a variant. There is a difference. Is a big change in the virus. Variant is a little itty-bitty change, and that's what this is. So it's important to keep that in mind. And all of the vaccines will work against this new variant.
Okay. Can RNA vaccines cause an active disease?
No. There is no live or attenuated virus, so the categorical answer to that is no, it will not cause disease.
And it will also not make your COVID PCR test positive.
Great, thank you.
That is correct.
Okay, so we've answered Bell's. So if we get vaccinated, if somebody gets vaccinated, do they still need to wear a mask and maintain social distance?
Yes. The answer there is yes. It's because efficacy, I think one of our speakers also answered that already. I think Professor Adeel was also emphasized a while ago that no vaccine is 100% efficient. It means that you still have to practice social distancing. And remember, there's one news, no, that a nurse was infected after 1 week of being injected with COVID-19 vaccine. It's because it takes a while for the vaccine to become effective. So I still promote social distancing, wearing of mask, and to implement protocols in the community.
Right. Great. So the next one is, so if you have an asymptomatic COVID-19 infection and you don't know about it, and without any PCR, you take a vaccine, is that okay? What kind of an impact do we see out of it?
So the answer to that is it'll be relatively safe. There are no safety issues that we can think of. What the immunogenicity would be, I think there are no studies, but we don't anticipate any difference based on what you know. And slight addition to what Faisal and others said early on, that if someone is PCR positive or has acute infection, we do not vaccinate that person. We wait for at least a month or so after the initial infection. There are two reasons for that. One is that the vaccine supplies are very low at this point in time, so we want to prioritize people who really need it. And second is that these people do have some level of antibodies which will at least protect them in the short term to a fair degree. Not as good as the vaccine protection, but they'll have some protection. So we wait for a little while if they are active disease or active PCR positive for a few weeks.
Okay. Because a lot of you are not enrolled, are supervising clinical trials. Have you seen anybody land up with a Th2 response to any vaccine?
No, not yet.
No, we've given 2,000 doses in my institution in the last 3 weeks and have not had any hypersensitivity reactions.
Okay,
we have given close to 5,000 doses and we haven't seen any anaphylaxis so far.
Great. So by that I mean none for Cancino, none for Pfizer, and none for Sinopharm.
We have only given Pfizer, so I must confirm that. We have only given Pfizer.
Okay. Has anybody been working on Sinopharm? I thought you were in the UAE and you might have some idea about
No.
Okay. So another question, when will interim results of Cancino trials be announced? Dr. Faisal, for you.
So for that, we need at least 50 cases which occur 28 days after the vaccination. We are about halfway there. So, and cases are coming every day. So probably in the next couple of weeks, we should have a first interim analysis.
Okay. And if the interim does actually show that it does have a superior effect as opposed to, obviously, you're not giving any drug in the other one, placebo, will there be a crossover?
There's no crossover. Then we got to wait for the second interim and the third interim, which is at 100 and 150 cases. Well, okay. 150.
Okay. Can vaccines be given to children?
So right now, the Pfizer vaccine in the US is approved down to 16 years of age, but there are no other vaccines that are approved down to that age. And the only trials that I know of are the Pfizer vaccine. I think that is basically enrolling 12 to 18 age group. So at the moment, no.
Okay. Adeel, you want to add something to that?
No, Wajiha is the expert on that. I'm sorry, Mehreen is the expert on that.
Okay, great. Another question, can vaccines be given to immunocompromised patients?
The answer to this,
yes, there is no contraindication.
Yeah, there is no contraindication. Yeah, I think there is no contraindication. They should definitely get it. Their ability to mount a response may or may not be as good as somebody who is not immunocompromised, but that does not mean that they should not get it. They should get it, and hopefully they'll get a response as well.
Okay.
I just want to add, it also depends on the vaccine, right? So the live vaccine we probably will not.
So Faisal, can you elaborate on that? Which vaccines can be given in immunocompromised?
So the Pfizer vaccine, the Mrna vaccine is a DNA vaccine. That's not really an issue. The AstraZeneca, the Sputnik, the Cancino, these have a live vector. So for this one, you just want to double check. And then for the Sinopharm and the Sinovac, these are inactivated vaccines, and that's fine also.
Okay. From among all the adeno vaccines, which one so far has shown to have the most promising results? And a little hint: is it the Russians?
So this is a difficult question. question because we don't have all the data for all of the adenovirus vaccine. The Russian one I know is finished, but we don't have access to the data as yet. They haven't published it as yet. The only one we have is the Chadox or the chimpanzee adenovirus, the AstraZeneca vaccine, which had decent results, I think.
Okay. Dr. Zara, do you want to add something to that? Especially with regards to the two, Sputnik has two particular spike proteins, I think, for adeno, 26 and 5, 29 or 5. I keep confusing.
So as Dr. Faisal said, we don't really have the data, so it's hard to tell without data and to compare efficacy of vaccines. One can hope that we'll have data on all of them soon.
So
well, we have seen press releases from the Russian government and the company that makes it, the Jamal Institute. They claim high 70 to low 80% efficacy, but that is based on a press release from the company and not on peer-reviewed data. Same is true for a couple of Chinese vaccines. They've reported from the UAE, for example, about 86% efficacy, again, from a press release from the government and not from peer-reviewed clinical trials. So that is all the information that I have, unless some other panelists have more.
I think the Russians are wrapping up phase 3, if I'm not wrong, but they did, I think 40,000 people were enrolled in phase 3. We're still waiting for the results as soon as they come out. Okay, another quick question.
I'm sorry, just to clarify one thing, the Russian virus vaccine does not have two spikes. It has two different adenovirus vectors at 5 and at 26. So two different virus vectors. And the reason for this is that at 5 gives you a more robust immune response, but there's a lot of pre-existing antibodies against this vector, while at 26, there's not a lot of pre-existing antibodies, but it doesn't give you as robust an immune response. So they're trying to mix and match both, so maybe hoping that they will get through.
Great. Another question for a post-kidney transplant patient, would we recommend vaccines?
Yes.
See answer above.
Yes.
All of the above.
All of the above. Okay. Do we check for antibodies? So in the trials in which you're recruiting patients for the Cancino and the Pfizer ones just completed, are you checking for antibodies a few weeks down the line, especially for an IgG long term?
So we are, there's actually a smaller cohort in which serial antibody testing is done. So not everybody gets it, but an antibody test is done at the beginning of the trial. The results are not disclosed, and at the end of the 1 year, and again, the results are not disclosed. Will be disclosed. If somebody comes with symptoms, their antibody response in acute convalescent sera is checked. So they are checked, but not for everybody, and depending on who you are.
So, for example, for the Mrna vaccine, about 2% of the people, if I remember correctly, had antibodies at baseline when they went back and checked. But the response rate was not different whether you included them in the final analysis or excluded them. The response rate or protection was similar.
Okay, great. Is there any particular reason why any DNA vaccine was not approved by FDA before? I know.
There's no DNA vaccine data that has been submitted to the FDA, to my knowledge, at this point in time.
Okay.
So if you don't submit to the FDA, they won't approve it.
Good. I think that's the best answer. Okay. So manufacturers say that effectiveness is 95%. Is that true for the Moderna and the Pfizer vaccines? I mean, are we going to suspect them or?
So there is no reason not to believe those data. However, the data that we see in clinical trials does not almost exactly translate into real life, real world data. And the reasons for that are many, because people in the clinical trials are carefully selected people, while in real world you have a much larger unselected population. Also, we have no guarantee if the cold chain will be maintained as per the manufacturer's requirement in real life and so forth. But if all those conditions are met, then I don't have any reason to believe that the efficacy will be any different.
Okay. Another quick question. Have we seen mix in adults?
So there is a condition. Yeah, so there is a condition called MIS-A, which is basically this MIS-C, but an older population, and it has been described. It's a little hard for me to understand whether that's just a continuation of severe COVID, or if it's true MIS-A, because what we're seeing is our symptoms 2 to 4 weeks after the initial infection. And I don't think that the MIS-A presentation from what I read is as clear-cut as that.
Great.
Agreed.
Okay. Another question. So I think Adeel did talk about this in his session. So alternate vaccine. So UK is permitting another kind of a vaccine if the first one is not already available. Thoughts on that?
No, we are not permitting that. We have a reasonably good supply chain out here. And the authorities in the US, both the NIH and CDC, do not recommend that. I know the Russians and the AstraZeneca people are teaming up to do a mix and match trial. But at this point, since we don't have any data whatsoever, particularly if they're two different types of vaccines, you're combining a vector-based and an RNA-based vaccine, we don't know what the responses would be, so we do not recommend that.
Great. Anybody wants to add something to that? So just in case, I mean, you guys have the supply, but if somebody doesn't have the supply, should they not? Okay, we'll skip it. Next one, travel restrictions. So UK has gone under tier 5 restrictions. How soon do you think they should be lifted from that and global travel resumed?
I think. Yes, I think we should depend on the cases arising in the country. Like in the Philippines, we just give a certain duration for now, but I think we will wait for the progress regarding the case rising in their country. So what we do here is we ask the travelers from UK to have a 14-day mandatory quarantine when they get here in the Philippines, so as not to spread the type of the UK strain COVID-19.
Okay.
And other,
and I think
other 20 other countries with the UK variant are also restricted with certain conditions of the quarantine. I think Professor Adeel would like to add something.
Yeah, I think that is an excellent strategy, which is what we employ as well. We have actually a tiered quarantine, either 1 week or 2 week based on where you're coming from, and you can either quarantine at home or you have to mandatorily quarantine in a quarantine facility hotel. We have designated facilities at all levels for that. And I think that is important. The other thing I want to mention is that transmission of infection in the airline, in the airplanes is very low. That has been looked at by certain groups. And we have been monitoring every traveler that comes into the country, into Qatar. And I'm on the government advisory panels and so forth. And the overall positivity in returning travelers is about 2% or so. And we have one of the largest airport hubs in the world where people travel through. So we have data on very, very large, hundreds and hundreds of thousands of people that we test out here.
And there are no travel restrictions in the US, and we have the highest number of cases in the world right now. So do not follow the US in this respect, please.
Just one thing we got to keep in mind though, and this is that we keep calling it the UK variant, but the only reason we call it the UK variant is that that's where it was discovered first. This may be elsewhere, which because UK does so much genomic work, so it's almost like the Spanish flu story. Where it wasn't really a Spanish flu, but they're the ones who were reporting it at that point. So,
and I think Faisal brings up a very critical point, and Zara would probably add on to that we found this variant in Colorado in people who had no connection and no travel history to UK people. So it goes on to tell you that these mutations will develop de novo, no matter where you are. It doesn't have to. It was not a clonal spread from a single source. So these mutations are probably evolutionary mutations in the virus that can happen anywhere, and I think more and more countries are seeing that now.
I'd like to add to that. You're absolutely right, because the primary clade that these mutations are arising in are the G clade, and the G clade is the most predominant clade in the world already. So again, it's wherever most isolates are being screened and where the strongest genomic epidemiology is, and being fed into a database where people are picking it up. So we are likely to see more as reporting increases.
Great. Another question, if a pregnant female gets the vaccine, and develops antibodies, will that be transmitted to the child and will the child be protected?
Don't know. Some antibodies, and again, I think Zara is the expert on virology. There are certain antibodies that do cross the placenta. I don't know if these neutralizing antibodies from SARS-CoV-2 will cross the placenta into the child or not.
So I did read one report where a COVID positive mother gave birth to a newborn, and the newborn did have protection, some antibodies. So we don't know enough yet, but it was just one case report.
And the same is for breastfeeding, I believe. Kids are not protected if they're breastfed by a mother who's developed antibodies.
Again, we don't really know, but we have been telling COVID positive mothers to continue to breastfeed because even if they don't have COVID specific antibodies, they have a lot of other antibodies and a lot of other nutrients that are absolutely essential to the immune system in a newborn baby. And so breastfed babies are protected in many other ways, and we would assume at some level against COVID as well.
Okay. So guys, we're touching 100-minute mark now, so I'm going to wrap up within the next 2 minutes. Another question that is being asked is the immunity going to be lifelong, or will we require doses like every year? I think Faisal did talk about it, but if anybody wants to add.
It's unlikely that the immunity will be lifelong, but it is likely that it will be substantial amount of time. Based on our experience from other coronaviruses, probably 1 to 2 years is what we are looking at. But again, this is a new virus. We have to wait and see.
Okay. One last question. Did this mutation arise as a result of jumping onto minks and then back into humans?
No. The Danish mutation is different. This is a different one. So the evolution of one. Let's not go there. It's there's some theories about this, but I think let's not.
I actually wanted to touch upon that because there's a lot of media hype on that. So we just want to negate and
Yeah.
So yeah, it didn't come from the main. It's not the same as the South African one. The South African didn't come over here. How this evolved is still theorized. There is a paper about the South African one where for the British one, there was a thought that this was probably an immunocompromised patient who may have had a prolonged infection leading to multiple mutations. Maybe somebody who may have gotten convalescent plasma or plasma therapy of some sort, where there's a skip mutations. However, having said this, in the South African clade, there's some now showing that maybe that's not true. So I think until we get more data, we'll just have to see.
Great. So I'm not going to take more questions. We have touched the 101-minute mark. So I am going to wrap this up. Thank you again, everybody. This was an amazing session. It was attended by a lot of people. We'll be releasing the recorded video soon. I really need to, thank you all. And we'll be, talking about summary points, and we'll get back to you in case we have more questions. Thank you, Faisal. Thank you, Dr. Adeel, Dr. Mehreen, Dr. Daisy, Professor Zara. If anybody has anything that we've missed in this session and you want to talk about it, now is the time. Otherwise, we will wrap up.
So one thing I want to add, I think that's a question in everyone's mind. We have seen no evidence so far that there's a Bill Gates, Microsoft chip in the vaccine. So we haven't seen that at all. There's no data to support that.
Good one. Good one. Professor Zara, do you want to say something? Any last minute words?
Thank you. That's enough from me. Thank you.
Great. Okay, so if no last minute words, thank you. Hopefully see you again. And thank you for bearing with us. It was a long but a very productive session. Goodbye from Getz Pharma, and hope to see you all again soon. Goodbye.